All right, so today we will tell you about the IGVF Consortium and the data resource that the consortium is building. And while IGVF is still generating data and predictive models, the goal of this workshop is to facilitate use of the resource by the broader community. So IGVF began in 2021 with overall goals of creating a resource to accelerate genomic variation research throughout the community and to provide insights into variants, genome function, and disease. And IGVF consists of more than 100 funded labs and other affiliates organized around several major activities with extensive collaborations, including relationships with other NHGRI-funded consortia so that our outputs are compatible. Overall, the consortium is addressing major challenges in genomics. The rate of variant discovery has increased tremendously through genotyping and sequencing in increasingly diverse populations, identifying more complex types of genome variants. And enormous numbers of associations have been identified between variants and disease, including common and rare diseases and related traits. But our understanding of how genome variation affects genome function and phenotype has not kept pace. So to learn how variants affect disease risk, several aspects of mechanisms need to be determined. Variants may function differently depending on the tissue or cell type environment. Some variants, for example, act through context-dependent regulatory elements to influence gene expression, some more directly on protein function, and some variants in genes can act in networks. And some effects may be detected as an effect on cell function, and others may be better detected in organoids or mouse models. Especially large challenges right now include the billions of potential variants, where it's not feasible to experimentally test all of them, the hundreds to thousands of cell types and cell states, identifying not just plausible associations, but causal effects, integrating across coding and non-coding variants and across disease areas, and building an infrastructure to organize, manage, and usefully share an enormous number of results within the community. Successful mapping of these connections could reveal insights into genome biology, guide diagnosis or clinical management of rare diseases, reveal genes, relevant cell types, and pathways that contribute to health and disease, and that guide development of new therapies. So IGVF aims to accelerate building of a resource with which a user could look up any variant and learn about its function or its predicted function in any cell type or environment. We have focused on generating data with validated experimental methods that's uniformly processed with well-calibrated output and using well-documented methods so that others can apply them. Our aim to have predictions that are benchmarked against gold standards and aiming for the catalog to be interoperable with other NIH resources. So toward the goal of understanding the impact of variation on function and phenotype, IGVF projects include single-cell mapping, genomic perturbations, and predictive modeling. And the projects use many different model systems, some of which are shown here. I'm now going to share very brief example projects to help explain the scope of data and predictive models that IGVF is generating. So first, as one example, IGVF is working to map the effects of non-coding variants in the genome. You'll hear more about that today. Key challenges are that non-coding variants in elements can have cell type specific effects and act over long genomic distances. An ideal solution would be a genome-wide map of which elements regulate which genes and which cell types so that we could look up the functions of any non-coding variant. To tackle this challenge, IGVF groups are mapping chromatin accessibility in hundreds of cell types and tissues to identify candidate regulatory elements, perturbing thousands of elements with CRISPR in a variety of cell types derived from human-induced pluripotent stem cells to learn which enhancers regulate which genes, and developing computational models to predict these CRISPR data sets from input single-cell data. Newer resources from IGVF include large CRISPR datasets in several cell type, benchmarking pipelines to evaluate predictive models on a common standard, and new models that use single-cell RNA and ATAC-seq as input. As a second example, genetic testing has identified over 1 million missense variants, but for the vast majority, we lack the evidence needed to classify them as pathogenic or benign. These variants of uncertain significance limit the applicability of genomic medicine. An ideal resource would enable lookup of any missense variant in any gene to learn whether and how that variant disrupts gene function. Toward this goal, IGVF is perturbing more than 200,000 variants in or around coding sequences, including saturation mutagenesis around certain genes where genetic diagnosis is clinically actionable, and deep phenotyping of selected variants across many more disease genes. These perturbation data are being combined with other computational annotations to develop new models to reclassify VUS. So these efforts have yielded new data sets, including a functional map of more than 9,000 variants in Factor IX, which has allowed reclassifying many VUS for hemophilia, and insights into how disease variants work. For example, showing that many pathogenic missense variants lead to mislocalization of the protein in cells. And as a final example, certain IGVF projects focus on studying particular diseases and traits. So one area is coronary artery disease and related lipid traits, where GWAS have identified hundreds to thousands of signals. The key challenge here is to link these variants to the genes, cell types, and pathways that they control to influence disease risk. To tackle this problem, IGVF groups have collected single-cell reference maps in human coronary arteries and cellular models, perturbed thousands of variants, elements, and genes in GWAS loci using base editing, perturb-seq, and massively parallel reporter assays, and are integrating these functional data with gold standards from human genetics to develop new methods to identify causal genes, cell types, and pathways. These efforts have produced resources to interpret coding variants in key genes such as LDLR and discovered a role for a specific signaling pathway in endothelial cells in coronary artery disease risk. So altogether, IGVF projects are in the process of generating a resource including single-cell mapping data in many cells in human and mouse, millions of perturbations to variants, elements, or genes, and predictions about the effects of these variants across many layers of genome function.